Hello, everyone, and welcome back for another week of Growing with My Fellow Growers. I'm your host, Jack Greenstock, joined as always by an amazing panel. I'm going to pass it over first tonight to Spartan Grown.
Hello, everybody. Thank you, Jack. I'm Spartan Grown. You can find me on Instagram, all one word, Spartan Grown, no spaces. Or if you don't do the social media, you can shoot me an email at spartangrown@gmail.com, and I can help you with all of your cannabis gardening questions. I'll leave it that way. It's the easiest way to say it.
Happy to have you back. Next up, we've got Matthew Gates.
Everyone, this is Matthew Gates. I'm an integrated pest management specialist. So if you're interested in that kind of information, you can check me out on my Zenthanol YouTube channel, also on Twitter and Instagram at SyncAngel.
Happy to have you back. Next up, we have Brandon Rust. You're still muted over there, Brandon.
Oh, sorry. What's going on, everybody? Brandon Russ here. You can check me out on Instagram at rust.brandon and Bokashi Earthworks and www.bokashiearthworks.com. Great to be here. And yeah, as always, it's gonna be fun.
Happy to have you back. And last, who's currently with us, but certainly not least, Noah, the grower.
How's it going, everybody? Yeah, I'm No Other Grower. You can find me on Instagram there, and happy to be here.
We're happy to have you back, and hopefully we got a few more popping in here in a little bit, because I didn't hear anybody necessarily say that they weren't going to be here tonight, but they might be unable to join for one reason or another that they weren't able to share, which is totally reasonable. Tonight we're going to be talking a little bit about what we'd call white paper or science research paper. This one's actually going to be a little bit of a meta-analysis I sent to our group about an hour before the show started. But I think that, like Tao and a few others in the comments mentioned, when we see it raw, it kind of gives us the first impression that a lot of the listeners are going to have because they're not super familiar with the paper. But this one is talking about abiotic factors and how they might impact production of like phytocannabinoid yields and things of that nature. So I'm gonna go ahead and share my screen, and I'll pull up the research article here. Oh, whoops. I'm gonna keep it minimized here for a second. Click over to the actual article. Here we go. Now we'll go full screen. All right, so it's Cannabis sativa L, crop management and abiotic factors that affect phytocannabinoid production. And so this was an interesting paper when I was looking through. It was done in 2022 in June. So this is very modern research. And it talks about the main characteristic of Cannabis sativa L is the production of compounds of medicinal interest known as phytocannabinoids, which many of us are aware of, THC, CBD, and things of that sort, but as well as terpenes and other minor cannabinoids. There's lots of interest in this. And so I figured it'd be interesting to go over. This actually isn't the full text, though. So we're going to hit view full text.
Also, for anyone curious, the L means Linnaeus. It's the taxonomist who characterized it first. So they give him the name there. Yeah, no, I've always found them to be kind of only really helpful when. Anyways, I'll let you keep going, Jack.
Well, no, I think it's important to note, but even like the word sativa typically just means cultivated. Like in garlic, you see like sativum versus sativa, but it typically characterizes that it is a cultivated variety, something that has been impacted or kept by humans throughout time, whether it's heirloom, landrace, or something that we've bred.
Though I want to stress very much, since you mentioned it, that just because a taxonomist gives something a name, it's not necessarily deterministic. So usually when that happens, it's for like crops and things that we've grown for a long time. So it does happen to be true, but sometimes it's not a relevant characteristic or even sort of a misnomer. Anyways, keep going.
So just in the abstract, it says, in this review, we aim to examine the current cannabis agronomic research topics to identify the available information and the main gaps that need to be filled in future research. This paper introduces the importance of Cannabis sativa L, approaching state-of-the-art research and evaluating the influence of crop management and environmental conditions on yield and phytocannabinoid production, including 1, pruning, 2, light and plant density, 3, ontogeny, 4, temperature, altitude, and CO2 concentration, 5, fertilization and substrate, 6, water availability, and presents or presents concluding remarks to shed light on future directions. So this paper essentially covers all of those different things, but instead of going over the entire paper, which we can look at the main findings, things like that, I guess it'd be interesting to sort of talk about the impacts of these different abiotic factors, and not to placate the audience, but the difference between an abiotic factor and biotic factor is like non-living versus living experience. Essentially, you've got, I guess I could just Google abiotic factor.
Well, it's like
give you a proper definition. But
well, I mean, I think I
yeah, you
can
can
do
take it
it.
over.
We don't want to spend time on it. Basically, yeah, like abiotic factors are like temperature, humidity, things that are not living that affect the plant. Biotic factors are the opposite; they're living things. I would even include viruses in that definition of biotic factors, and microbes and insects and mites and I think I would also even include I might use a different term, but the plant itself or other plants in tandem, like in a crop or something. Those can have effects on each other too.
I found it interesting that they lumped so many things together in a particular one, number 4, temperature, altitude, and CO2 concentration. I guess when you're looking at like natural cultivation, all 3 of those things are going to be included because based on the environment that you're cultivating, you're going to take those measurements. So each cultivation site is going to have those factors kind of all uniform essentially per that location. But I feel like they can be examined individually, like temperature, altitude, and CO2 concentration. Like they could have been 4, 5, and 6, and then 7 could have been fertilization.
Well, they probably are separated. Oh, but you saw the paper, right? So I mean,
yeah, it's I guess they examined a few different papers that I have read through it, and these 3 are typically, because they're looking at like outdoor cultivation sites. The thing that I guess we could jump through, the introduction, there's a lot of just with any introduction background information about like the history and issues of research and lack thereof.
I think one thing to highlight in that though, Jack, that a lot of people aren't aware of is, and it highlighted in there, it's only maybe one sentence, but they're talking about how the United Nations has even, it's on the second paragraph there, how the United Nations has even downgraded its list on the dangerous substance list, and the United States is still behind the United Nations. What the fuck is going on?
Yeah, I think there's definitely some politicization that's going on there, and a lot of the rest of the world, whether it's Canada or many parts of Europe, have moved forward with cannabis and allowed research and things like that to happen where we currently still have it federally illegal. So with that aside, I do think that this examines a lot of research from countries that actually have a little bit better policy towards research and just understanding cannabis. So there's lots of different things that they've looked at, including CBDA, CBGA, THCA, and just decarboxylated THC, which many of us know and love. So I think they even discuss the difference between pollinated by an individual plant versus like pollinated by many plants, and the impact that might have on phytocannabinoids and terpene profiles and things like that. It says there is a misunderstanding that cross-pollination changes the chemotype of the plant. However, this change only appears in seeds resulting from the cross-pollination and not in the pollinated plant. So I found that to be a little bit interesting, at least in this research. That is what they're claiming. But to get down a little bit further into the paper here, for those who don't know, a meta-analysis is looking at tons and tons of different studies and pulling all of the findings and kind of trying to draw conclusions from lots of different research. It's not one individual study. So this is looking at studies out of Canada, Israel, and some states in the EUA. And so there's many different articles that were highlighted. And I guess this is just sort of to highlight the fact of a lot of us think of the biotic factors, like the living things, the microbes, the people that are actually cultivating the plant, and the other biotic factors, like pests. Matthew deals a lot with pest management, molds, mildews, viroid, things like that. So it's important, I think, sometimes to acknowledge the abiotic factors and how important they might be. And there's a ton of research actually going on around those, because many of them are not easy to measure, but measurable. And scientists will take the time to go through actually measuring all these different things. So just continuing on, they talk about management techniques. Like one of the abiotic factors they were talking about was pruning. and also environmental controls such as stress conditions, nutrient deficiency, competition, such as like planting too many plants in a single area with other plants, reduced water availability. And they note that that could increase phytocannabinoid production, which I think that they're referring to the University of Guelph study that we've mentioned on here probably dozens of times at this point. But it is interesting to see that they're considering that as part of one of the protocols. Sorry, I'm not the world's fastest reader here, but I did research this. I should have given everybody a little bit of time, I guess, before. Here's a little bit of a figure, and it's talking about some of the possible effects on yield that you could have by pruning, and the impacts like it has hormonal changes, manipulating the morphology, better light exposure, better air circulation, aid to pest management. These are some review for a lot of us, I'm sure, but it's good to see scientists noting and acknowledging this in modern research so that it gets pushed out there to other researchers so that they can do some more proper research. Because I have another research paper that I didn't share with the group, but it looks at like different ways to treat water and how it impacts plants. And it was amazing again to look at the unhealthiness of even like their untreated water control condition, to see like their healthiest plant was okay at best, and all their other plants in the experimental controls looked like they're literally dying, like complete garbage. So it's nice to see some good information at least being publicized for the research community out there. And as a whole, people are moving in the right direction generally, I think. I'm going to close out this little diagram here. And if anybody has questions or comments or wants to jump in, I should probably also double check to make sure. I'm going to hit stop share for a second.
Right there. Right where you were at.
I'll reshare because I thought somebody maybe jumped in to the Zoom, and I didn't want to have a panel member like waiting in the whatever waiting room, not being able to join the call. But go ahead, Spartan.
I like that in the it's cited as 73 there at the end of that third paragraph, looks like down. It says that the technique known as lollipopping did not influence the productivity of flowers or cannabinoids. That's the bottom of that paragraph where you started to highlight. And like I could see some people take that out of the subject, the last sentence in that paragraph that you first started highlighting. I could see someone take that out in context and say, oh, we don't need a lollipop because it's showing it doesn't increase phytocannabinoids. Yes, but like with this paper does actually say this, I think, in a little bit later on, but it says that there's 3 reasons why you would do plant manipulation, and not just one. It could be to increase phytocannabinoids. It could be to increase, I think they just said what we say, increase airflow. I can't remember the words they used. Airflow through the canopy, I believe is how they said it, or to increase the yield. So this is saying for one of these things, lollipopping didn't show a correlation to being positive or negative. So this isn't saying lollipopping wasn't effective. It just says it's not effective for increasing phytocannabinoids.
I think that's a good point to make. And that at the very top of the paper, I kind of noted that they rightfully acknowledge that there's a lot of further research to be done. This is a lot of the early research, and it's kind of accepting or a conglomeration of a lot of different research. So it's giving us an overview of many of the different things that are being done, as opposed to just picking on like one particular study, which is also fun to do, and we can do either later tonight on the show or in future episodes. I
do have one if we want to transition at some point, ready to go, but take your time, no rush.
I'm just trying to get down to the results. It's a lot longer than I actually on my computer scrolling and talking through it than when I was reading it in my mind on my phone. But plant density is another thing that they talk about. Competition generally is one of the things. Ontogeny is something that is newer to me. I haven't heard talked about too much in the community.
Ontogeny is how it came to be, basically.
Yeah. Jack, can we pause? This is the last time I'll pause you, but can we pause you and go back up a little bit where it's like 3.3, the paragraph before 3.3 plant density? This was kind of a interesting thing to me. The paragraph before that, yeah. I think it's like the first or the second sentence. Basically, it's saying veg length, and they're saying shorter, suggesting here shorter veg length is better. Does have a difference on the phytocannabinoid yield, and that if you do a longer veg, you might be hurting your phytocannabinoid at the end.
Well, I
thought
remember
that
Noah
very interesting.
Vigroa said this like years ago. He's like, the little baby plants, the short veg, produce the dankest bud sometimes, or maybe I'm misremembering, but I feel like Noah has said that a long, long time ago. So it's interesting that maybe there's some truth to be found in their research. Noah, do you remember mentioning that, or did you ever have that kind of thought in mind?
Oh yeah, no, yeah, I agree that. I just think there's a point that eventually, like if a plant gets too big, that it can just, I don't even know, like maybe the root ball can't support it. Like, there's just nothing science. It's just from seeing different plants. And I think there's also a happy medium, but you're right. The little plants, so long as they have the proper, pot size for their, for that root ball, they can produce pretty well too. So, and it seems like also like, it seems like the vigor of the plant of the flower is a little bit better when you don't stretch it out too much. That's just my experience.
Some of my dankest buds have been really short veg times and then quick flips on a tiny plant that just put out some amazingly dank buds on like what looks like a dwarf plant or almost like an autoflower, and it finishes in almost a similar or faster timeline. Like if you do a 30-day veg and cocoa, flip to flower, and you have a 64-day or 65-day flower period, and you're under 100 days, which is faster than we had Dog Doctor on, and he had a 100+ day auto. So from seed, you can get photo period to have dank bud yield in under 100 days. And it might even be, according to some of this research, some of the best stuff. Like that might be the way to go about doing it is, and I also kind of, because I saw people having success with smaller plants, I stopped vegging as long because one, I have a small place to grow in, but two, I hate lollipopping like having huge naked legs, like where I see people, half of their plant is bare stalk and the other half or less is buds. Maybe there's like only 25% of the plant left is producing buds, and the whole bottom like 50 to 75% is naked. And I think that would be better spent as a shorter veg time and just allowing the plant to, grow and have more buds on it. But it really comes down to everybody's preferences and cultivation environment. But
I remember on the previous paper that we talked about, they had mentioned that although their research report looked at like, 8-foot tall plants, right? They said way in the beginning of the intro that smaller plants, have a lot of qualities that make them better for exactly what we're talking about here. And I don't know like the why of it. I'm sure it's a question. There's multiple factors, but they did mention like the plant hormone nature of like how basically there's a lot more communication like time right between like the bottom and the top of the plant in different parts. So if everything's smaller, it's kind of like all the reactions are compacted too, kind of. And I think that makes a lot of sense, but it's not super intuitive because like with other plants, you get them big and strong, you expect to have like a good fruit harvest or something like that. But that's not always true either, right? So it's sort of like you got to give them a little bit of that stress to some degree. Of course, that's a loaded word, but like you said, plant manipulation, right? Like maybe if it's smaller, maybe if you're like, you don't give it as much time to prep, and then you, of course, kind of anthropomorphizing the plant when I say it that way, but you
it's
give
a living
it that.
thing.
It's a living thing, yeah.
Living things do have responses to stresses.
Right, right, exactly. And so perhaps like it's like it's like if you let the butterfly come out on its own, it has to pump in those fluids and get those wings up. If you free a butterfly out of its chrysalis, then that really important factor doesn't happen, right? And so then you have, at least in some cases, anyways, its wings don't develop and it can't fly. So I think there's a metaphor there.
For sure. And I think for those who are podcast listeners, this might be a episode where you want to check out the YouTube, because I'm not going to go through and read off these entire titles, but the sentences that Spartan was just referencing has 5 different research papers associated with it, and they're all cited: 102, 103, 41, and then we've got over earlier 67 and 87. And I've hovered over them, and it'll actually show you the authors' names and the titles of the research papers that they're citing in this meta-analysis. So if you want to go and look deeper into any of the claims that are made in this paper, I'll share this link on our YouTube after it's posted, as well as you can go through and just Google some of these names when you hover over it, like Lenton S, Frank, etc. And you can go through and check out the papers that these claims are coming from. So just as a reference for anybody out there who wants to do that further research later on. But yeah, moving on a little bit, we got the plant density. We were talking a little bit about that earlier. There's lots of claims. I think Brandon probably has some experience with this lately, growing in beds. And I'd be curious, just as we go on next to ontogeny, before we get to that, Brandon, what are your thoughts on like plant count per square foot? And generally, do you have any thoughts on that sort of abiotic factor, or how to optimize getting the right amount of plants in a bed so that they don't outcompete each other or have mold or things like that?
It's a combination of both what you're after as far as your production is concerned, because, well, for instance, in beds, I would prefer to do short veg time and do more plants per square foot. Doing 12 to 16 plants in a 4x4 area and only vegging them for maybe 5 days or right until they're acclimated into that bed, and then just flipping and then doing the plant maintenance that's required for that dense of a canopy. Because what you would be looking at like that is you wouldn't be topping anything, right? But then it's all about like the labor, what you're after, and trying to, for me, maximizing that if we're talking about operating at scale. So if I want to get more harvest per year out of a room, but I'm running beds, I need to have a short veg time. So I could do more plants. And if I did that, I don't have to top. But if I was going to do more of a veg time, I would have to, do more plant maintenance in veg. I'd have to top them, branch them out. I would do less plants to get the same thing, but it would require more maintenance on the front end, like in the veg room before they go into beds. You know what I mean? So it's just you have to take all those different factors into consideration, because every different cultivation facility is different. And that's one of the things as a consultant, right? So it's not all people operating in beds either. Some people are doing this in fabric pots. And so every system is a little bit different, and you have to take that into consideration.
Well, I actually just read a great paper that maybe we'll go into this later. It was called The Case for Micro Cultivation and Local Cannabis Cultivation, because as we're seeing the increase in larger industrial scaling, a lot of issues come along with that. And in many ways, I think the local markets often, whether it's, boutique agriculture or whatever, horticulture, can be served locally better than they can on a national scale many times by preference. And so it's one of those things that sometimes the smaller scale can actually do a better job than the large scale. And maybe many people would argue that's always going to be the case as far as quality and consistency or artisanal type things like that. But the plant density thing is definitely an interesting one. I'm going to pass it to Spartan, I guess, for a second, because I know that you have a plant count at home, and then at Mitten Canada, I think you guys also had a plant count. And what's your general thought on plant density and how it would impact cultivation and phytocannabinoid yield, or just yield in general of dry flower?
Well, I think that your question kind of gave the answer right away. It's like, yeah, that because we're in a plant count state, it kind of ties our hands into growing bigger plants if we want to get bigger yield eventually. If you hit all the other numbers, the only other thing you have to do is to get more yield is to grow a bigger damn plant. Because we're limited by plant count. If we were limited by canopy space, we could grow much smaller plants and just have a lot more plants. But so that, I think, is the only reason why we're growing larger plants. And I feel that if we were under a canopy size restriction, then it would be smaller plants than what we grow. I mean, there was days that normally I would say the average plant is a little over 5 foot tall, but I would say we've had some 6-footers in there for sure. And but I think the more ideal plant would be like a 4-foot tall plant or a 3-foot tall plant. But as you get smaller, you add more plants to fill the space, and you lessen the veg. Which
I'm with you on that.
As this paper kind of says, it's like It seems like when you have the more plants too, that like if you have one plant that struggles, it's not as big a deal as if you have large plants. It's more of the putting all my eggs in one basket kind of a deal.
Yeah, you can cut out a tiny plant and it's no issue. Versus if you cut out a 10-pound, like a Mendo dope, if they lost a plant, it'd be a fucking huge deal, right? Because they are such a monster.
Yeah,
So-
I want to also reiterate that, just not to belabor the point, but I think there's one additional reason why people grow plants larger. I mean, there's others really, but one main reason is that, like I said earlier, it's kind of intuitive, right? You grow a big plant, you get big volume, you get lots of all the other things you want, right? But no, not necessarily. And so I do think that there's a large swath of people who fall into that sort of intuition trap. Certainly when I would think that as well about a lot of things, especially like I've said in the past, when I was first entering into this space more professionally, I'd read a lot and written a lot and had learned a lot about this already. But the fact that you could even have massive differences between varieties, right, was something that I knew intellectually was true, but didn't appreciate as much until I started to work in a more commercial setup and sort of see all of those different factors, especially like with tomatoes and gerbera and things like that. And so yeah, that's my little input there.
I also saw another paper just to feed anybody's curiosity who likes to look into the stuff either now or later. There was one about grafting and how it increased phytocannabinoid yield. I think it was for CBD plants in like a one-step graft, and another one that we may touch on either later tonight or another date. But Spartan, I think it'd be interesting to see if Mitten Canico all of a sudden, let's say Michigan for whatever reason lifted the plant count thing, and then they said, hey, it's canopy now. And then they started saying, all right, we're going to go a little bit smaller and veg our plants a little shorter and flip a little sooner. I wonder, because you guys have a pretty long legacy of cannabinoid testing at a certain lab. So let's say you had that Donnie burger that's hitting 40-ish%, and then you start vegging it a little shorter. I wonder if it would creep up to 41, or maybe it would drop down to mid-30s, and then we could have a little bit of our own research to compare.
Well, at the same time, what they didn't divulge in this was they were looking at phytocannabinoid only, and they weren't looking at terpenes. So was kind of, I think, I'm hoping the market will move towards is wanting to know more about terpenes as well, because I think those really modulate the high more than the cannabinoids in some aspects. So
here in Oklahoma, it's required to have a terpene test for your product.
That's awesome. I wish they would do that.
So it has to be on the package and everything else, just like the THC percentage.
I like that.
Soon enough they'll add esters too, but I think if you look at wine, they only list the alcohol percentage, but a sommelier will be able to tell you, oh, this one's going to taste more tannic or more bitter or more fruity or more leathery or chocolatey or grapey, whatever it is. And I had a bottle the other night with my wife, and I smelled it and I go, oh man, this is boozy. I bet it's probably a 14 or 16%. And I was fucking wrong. It was 12%. But the volatiles, the tannins, and the other things in there, like the terpenes or whatever it is, that was making it smell kind of like boozy, even though it wasn't a high alcohol percentage, it was a red blend, and it was a bunch of bolder California reds that have kind of a very strong nose. And I think with cannabis, we'll start to see similar, even if it only is sold with THC percentages, we're going to start to see the super high potency stuff and the mid-grade potency, because a lot of people, old school smokers, actually can't handle some of the more modern stuff. They get panic attacks. They don't want to smoke it. They maybe want a little bit of CBD, or maybe they want a 15 to 20% or not a 40%, because it's just too much potency. So it's definitely a interesting proposition, because is max potency even the best thing? And one of the other things I failed to mention, even though I do think that the lab testing is getting better and better, they've only got 40 labs here, and they shut down 10 of them, and they're really expensive to get. So I don't think that people are cheating them like people think. But one of the things that people are doing that I found out through my wife, who works in the legal market, is some of them, I thought, oh, well, they can't just infuse a single bud and then send that bud in for the test. But some of them will take all of their keef from their trim trays, and then they re-infuse all of their buds. So they're essentially selling infused buds as non-infused, because it bumps that 30% up to 35, or 35 up to a 40, because they're just throwing keef into the total batch. And that was kind of a tricky method. I think that might be why we're able to see these higher percentages coming around, even though testing is a a lot more stringent than it's ever been. But moving on, I guess, unless Noah, do you have any thoughts on the plant count or plant density before we move to ontogeny? I'm going to take the silence as a no, maybe, from Noah the grower. And I'll pass it over to Matthew for ontogeny, because this is one that I'm definitely less familiar with. And it sounded like you wanted to jump in there when we started talking about this earlier.
Yeah, sure. So yeah, I mean, they say it right here at 3.4. First sentence reads, ontogeny is the entire sequence of events involved in the development of an organism, right? So like, you know, when you develop as an embryo, right, or as a zygote even, you know, all of these aspects of development that happen, you know, in that span of time. And you might even include other factors before that period of time, but you know, usually we're talking about a sort of developmental stage. So it starts from seed and goes through different developmental stages, such as the seedling stage, the vegetative stage, and reproductive stage, and ends at the senescence stage, right? You know, I didn't read this article earlier, but yeah, I will reemphasize what Jack said. There are, as well, a meta-analysis should. There's a ton of research papers that are referenced here at the end, you know, the third sentence from the end of this paragraph. And they do say there's like 1, 2, 3, 4, 5, 6, 7, 8 studies that are supporting this one statement, which is that the concentration of phytocannabinoids changes with the development of the plant, which is pretty intuitive.
Seems like a platitude, right? But they have to have
Yeah.
8 studies to support that, which is kind of crazy. But it just shows, look at how they say cannabis isn't researched enough yet, which is one of the biggest arguments against federal legalization. But like, look, they have 8 studies to support the most simple statement, which means there was 8 research articles all done just to be able to support in a different stage of growth. Like, oh, at seedling stage, there's one type of cannabinoid. At veg, there's a different type of cannabinoid. Flower, late flower, early flower, it's all different.
Explain to me how they, like, I knew that intuitively, but then when I hear of companies that will offer services to breeders, for example, and they will take a leaf tissue sample early on in veg and be able to tell you that, oh, this is a high THC plant, or this is a high CBD plant, or how are they able to tell what the final breakdown is going to be if it's changing throughout the life cycle?
Well, it's an estimation. I think what they do is they look at some genes. I mean, I don't really know in great detail, and I'm sure it's different for different groups, but yeah, they're probably taking you know, a look at the genome maybe and seeing like maybe there's some single nucleo
there's markers. So there's genomic
markers.
markers. There's a guy, Reggie, from Steep Hill Labs. He is a genomicist, and he's been doing cannabis testing since the early 2000s. And they have terpene analysis as well as cannabinoid analysis for thousands of different cultivars that they have stocked up in their banks or whatever. And then as soon as the genomics started coming around, they'd be able to say, okay, here's all of our analysis, because like in Prop 215, since 1996, I want to say, in California, we've had legal medical cannabis, and a lot of it started getting tested from then till now. And as soon as genomics came around, they started tying certain markers. Like I was really interested in pinene, and he was saying that from like how a Farmer Freeman test, you could take a cotyledon and or the first leaf sample and send that in, and they'll be able to say from the genomics that based on, you know, these other hundreds or thousands of cultivars that we have had sampled when they're grown out all the way, that there's a potential that it will produce this, and maybe this one never produced that. Like if you're looking at like OG Kush or something, it might produce certain terpenes and esters and flavonoids and cannabinoids, but then certain things maybe just never showed up, even in hundreds or thousands of tests of that across several different grows. So
Also, oh, sorry. Also, I just want to say that, yeah, like it's them putting all these studies here are not to be like, see, guys, we knew it was controversial, but here's the research. No, it's because as a matter of course, when you do this kind of thing, you want to make sure you're supporting your statements, and if a statement happens to be super well supported, well, you should probably do that, especially in a meta-analysis. So it's not like. I just think it's really important that people know that the reason that they might be so aggressive with their citations is just, you know, it's just good academic praxis. Because
myself, I've been critical of science lately because it doesn't replicate. What does this show me? You've got 8 things that all replicate. These all 8 of these things support the same statement. So they went into research and they figured out that at different stages in development, there's different phytocannabinoids. And I'm not going to go and spend a whole bunch more time to try and disprove these 8 articles. I'm pretty sure they all tried to disprove each other and then found out that the other people were correct. They found similar findings, and that's called replication. That's how science should work. In other labs and other settings, they should find similar things if it's actually a scientifically observable event. So I'm happy to see things like, 8 researchers, and it's not just one researcher. It's like, if you look at these, they're teams of researchers that spend years on these articles, and tons and tons of money and time and lab analysis goes into this at some of the highest levels of science that we have available to us. So
I do like that you're putting. I love that this paper is set so that you can mouse over the links and then see the authors and the cited title. But can you click on table 2? So under here, well, but like you see how there's, okay, that works out. Yeah. So here we can actually see some of those reports. A non-exhaustive summary about the most relevant studies in relation to the effects of ontogeny on phytocannabinoid production. So, and so on the left here, for those who are watching visually, there's the objective, which is the objective of the paper, and then we have the results of the paper. So, like, for example, the first one, the objective is to evaluate the evolution of major phytocannabinoids during cannabis growth. And the results for that, maximum concentration of THC-A and CBD-A in flowers were attained at the end of flowering during senescence, but varied according to chemotype. Now, that definitely tracks, but, that is, so it's important to know what they were looking for too, because then sometimes it might make sense to you, like the viewer or the reader, like, oh, why didn't they include this? And why didn't they include this? And I wanted them to take a look at this too. But that could have very well been outside the scope of their resources or their objective. So you got to have something that you're looking at or trying to figure out when you're doing these. Sometimes you can have a big paper, but you don't always have to have such a massive scope. And scope creep can be really expensive for research. So
I thought it was funny. They sort of flex in saying non-exhaustive summary because they're
way
like,
more. Yeah.
Yeah, we got a whole lot more. This is just a little bit. And this is a lot. I mean, we're looking at 1, 2, 3, 4, 5, 6 claims with the objective and the result, which is cool to see wrapped up so neatly. Here we've got investigating the impact of harvest time on phytocannabinoid yield. And we were seeing dry weight of the plant is increased at the 5th week, and there's a maximum at the 11th week. So that must have been in a certain cultivar. I'd imagine it's definitely going to change cultivar to cultivar, but it's worth looking into. This is number 72, and I just hover over it in there. If you want to press pause on the YouTube when you're watching this afterwards, you'll be able to go through and check out the reference to that article. So yeah, I love this type of being able to look through some of the research kind of quickly and get an idea of where we're at, like where we are in 2022, because the research changes every single year. We get more and more information, and it's kind of interesting to see where the scientists and researchers are at, because a lot of us have similar ideas, or maybe we have different ideas, and trying to figure out how to get them to line up and see who's ultimately right in the long run.
Use your filters when you search. Like Google Scholar, for example, has a filter for within the last 10 years, within the last 5 years. That really helps you on your search.
Absolutely. Want to emphasize that.
I just did 2022. There's an option you can literally do this year or last 2 years or last 3 years or whatever. And I think a lot of people, I've been guilty of this myself. I'm like, oh, Tal, that paper's from the 1970s, dude. Come on, give me something modern, because I have reason to believe that since then, maybe our methods have changed, maybe our technology's gotten better, and there's so many different things that, and maybe there is some landmark studies from the 70s that are still very, very solid to this day, and they're still quoted and referenced. But I think
sometimes they don't even. Sorry, not to interrupt. Sometimes they don't look. Sometimes they don't go back and look or redo it, because it's sometimes it's because it's sometimes it's because it's very, very demonstrably proven in that paper, even though it's old and there's nothing about the fundamentals that they're looking at. Sometimes that's not true, right? But sometimes I've noticed that too. Sometimes I'll be reading a report and it's the most recent example of this you have is the 70s. All right, cool, no problem. Also, Spartan, what are you having? What are you taking? What are you using to slake your thirst just now?
That was peach iced tea. That's pretty
Nice.
damn good.
Very nice. Very important to know how you're hydrating.
Always,
The
man.
fans want
Always.
to know.
They might not be able to see.
Actually, I do get asked often, honestly.
I always think it's a beer because it's always in like a big frosty mug. But then I'm like, so I don't think Spartan's a big drinker. And then I'm like, oh, it's tea. It's something probably healthy.
Yeah, it looked like it could be a dark brew. I was like, all right. That's a pretty big that's a honkin' Steiner you got there.
Man,
So
it's glass, man, and I got it at the dollar store. So I got it for $1. I was like, hell
that's
yeah.
not bad.
I love the dollar store for stuff like that, or like whatever the Goodwills or type things like that. But we've got.
So what else do we got here in the ontogeny section?
Evaluating the concentration.
Oh, besides the. Oh, I meant like after or like
okay,
aside
after
from the
table.
table.
Let's see if I can click out. There we go. Good. It didn't make me go all the way up to the top. They have a whole lot actually, just like earlier. I mean, we've got probably at least 24.
Ooh, look
articles.
at this! Look at this stack at all on the top of the third paragraph here. The citation 110 says here, Stack and others observed the rapid accumulation of phytocannabinoids in different varieties about 3.5 weeks after the onset of flowering. Hamami and others in 2021, both of these were in 2021, evaluated the phytocannabinoid production of 12 varieties intended for fiber or grain production, so that's important context, at different stages of the reproductive phase, and observed that the synthesis of these compounds varied among the genotype and during cannabis ontogenic development. So I guess it's early development is the way that I'm interpreting that sentence.
I'll say intended for fiber and grain production. We can just go ahead and say that's pretty much describing hemp, right? This is not
I'm pretty sure.
High
probably
THC.
Probably like phenol. Yeah, right. So that's like the important caveat is that that's what it was for. But like, there are some fundamental similarities that might be there, or that you might expect from other plants with that have some of that heritage mixed in. I think
I've been guilty, I guess, of trashing on hemp, but like I'm usually referring to the old school hemp, like the classic hemp that is very low CBD and very low THC. It's actually meant for fiber and seed, where now we have hemp being 0.3% THC or lower, and there's a categorization of hemp which is really like medical cannabis that is just very high in CBD.
It's not marijuana, quote unquote. It's
totally not marijuana.
It's don't put me in federal prison. Marijuana.
Don't smoke me, bro. Even though I'm legal in all 50 states. But I think that category of hemp has a lot of medical, therapeutic, and especially commonality between cannabis, and it's much more similar to cannabis than like the fiber or seed hemp of old. And even modern fiber and seed hemp, I think, have a little bit higher CBD levels than the older stuff. So it's a much different thing than it once was. And even though hemp is looked at as a different plant, it is still cannabis. So these studies often do have a lot of overlap that is relevant. So I'm glad that they include them.
Let's scan down.
First 3 weeks is interesting because I would have felt like the cannabinoids would have been packed on a little bit later, but I'm sure that they're seeing just like a rise from pretty much nothing to now it's existing and on a pretty steady incline.
I was confused with this next paragraph right here that starts with indeed the concentration of the phytocannabinoids varies. What do they mean by photoperiod sensitive
phytocannabinoid?
Neutral. What's the difference between a photoperiod neutral, photoperiod sensitive? What are we talking about?
So sensitive would be, neutral is autoflower and sensitive is photoperiod. It's photoperiod, right?
Yeah, it needs the darkness to turn flower. Where neutral would mean it doesn't matter what the light cycle is like. It doesn't care what the light cycle is like. It's going to flower when it's mature enough.
You see what it says in this? It says that it's saying that, okay, so autoflowers, the CBG concentration starts out high in its flowering life cycle, but then in the 12th week, it comes back up to the maximum level. What? That's
so I think what they mean is that I had to take a look at the paper, but perhaps what they did is they took samples at different stages of life, and they saw that like the levels of certain cannabinoids like fluctuated, right? So it was like higher at this level and then
So they
dipped
just meant
down.
12th week of life, not 12th week of flowering period. Because I'm like, what autoflower
I
goes
don't.
12 weeks?
Yeah, I'm just speculating based on what you've said, but I haven't actually in detail read it.
12 weeks of life, I would imagine you're right. Yeah, because like
Yeah.
2 weeks or 3 weeks vegging and then 9 weeks of flowering.
Oh yeah. These are academics. They're probably not meaning 12th week of flower as a presumption. Maybe.
Well, they're just
but
going to
I
count
think
weeks because if
I
it's
think you're right.
neutral, then they're just going to count weeks of life,
Yeah.
and they consider it kind of
Yeah.
like
It's just
it's
in
always
that-
flowering in their mind,
just-
even though there is a small vegetative phase.
Why it confused me, it was just in that sentence right there. I mean, in that it says it starts out as in the photoperiod neutral genotypes, the maximum concentration of CBG was in the second and third weeks of flowering. And after this production peak, a significant drop in CBG production was observed, but in the 12th week
Of flowering, I guess. Yes.
so that doesn't make sense to me. It's
Oh,
like,
I see what you're saying now.
So this is from a study of, again, we're talking about, it says development of industrial hemp. So the CBG amount, because I think it can only become CBG going to CBD, and maybe it's not going to CBN, it's converting back to CBG for some reason, or it's producing more. Maybe it's like re-vegging would be my philosophy potential or theory on that.
I think that they're just taking it all the way. They're just like seeing what happens. I don't think that they're doing it to like they're not as like a farmer would, but as just a person who's like, we'll just keep it living and see how it goes. Unless, I mean, I guess that still doesn't make sense because that's still kind of a long time.
I will say it's interesting that, just like you'd find in most modern hybrids, they're talking about the maximum total phytocannabinoid concentration in photoperiod neutral varieties was 8th and 9th weeks of flowering. So it's almost like just like a photoperiod, but they're saying photoperiod neutral. So autoflowers are most potent at 8 to 9 weeks into their flowering cycle, just like
I'm not convinced that
photos would be.
photoperiod neutral means autoflower because it doesn't make sense in that context to me at all. I was so confused by that paragraph.
Could you have like a long? Yeah, that is sort of odd. I have to admit. I guess we'd have to take a look closer. But
I could click on the article, maybe. Let's see. Google. Oh, wait. Oh no.
yeah, no. Click on the go up a bit. It's 120, right? Yeah, young.
Yep, here.
Let's just take a look. Let's just take a look.
Development of cannabinoids in flowers of industrial hemp, Cannabis sativa L, a pilot study.
3-day length sensitive, 2-day length sensitive neutral varieties.
Day length neutral variety. That sounds like autoflower to me, but what autoflower goes 12 weeks and
go up actually back to the abstract. It says here, I noticed this key word here. They're saying results indicate that total THC, CBD, and CBG significantly increased as flowers matured, reaching the greatest concentration during 6 to 7 weeks post anthesis. So post flowering. After a plateau stage of varied length for different varieties, the peak concentrations declined as plants senesced.
And you see that here with the diagram. So look, it goes up, it hits a maturity point, and they're saying 6 to 8 weeks, and then it starts to drop down after that 8-week period in more total. And it's similar to like a lot of people feel like if you don't harvest your plant when the trichomes are milky, if it starts to get amber, you're going to start to see decay from THC to CBN. I really think a lot of it is the terpenes changing and things like that. Because photoperiods, you have to run really, really, really long to see any CBN. Like I've looked at thousands of tests and I've never seen flower with more than 2% CBN, and that was the highest. It's usually like less than 1%. But with this, we're seeing pretty drastic drop off after week 6. And so these autos seem to be finishing fast. I don't know if we have the full text here or not.
If they were CBD autos, that makes sense because you want to harvest it early, I would imagine.
They are CBD
I think it's
autos.
I think because they're hempy and they're like, I think that this is sort of
Well, and what I know about CBD 1-to-1s with THC is the later you wait, the more THC comes around. So CBD is earlier in like a 30-to-1. Harlequin, the guy who found Harlequin, House of Harlequin, Wayne Laughter, I think is his name. He said it's a 30-to-1 if you harvested it like week 5 or 6, but if you wait until weeks 7 or 8, it becomes like a 16-to-1, and if you push it to weeks 9 and 10, it becomes like a 4-to-1. So predominantly CBD in all of those ratios. But the fact is, the later he waited, the higher the amount of THC went up. And in this case, it looks like, well, they are even showing THC in this as well, but that seems to drop off. So it really depends a lot from cultivar to cultivar. This one is an interesting one because, again, they're looking at flowers and industrial hemp. So I'm not sure why. We're looking at really low levels. Wow. Yeah, THC percentage, we're talking 0.4. So you went from 0.6 to 0.4. So that's not like a huge drop off, right? And CBD is more of a drastic drop. You're going from like 12% at the peak, and then the valley is like an 8%. So 4% drop off is pretty significant there. Or it seems more significant. I'm sure statistically it's significant because if you look at the graph, the THC is dropping a similar amount. It's just in such a low quantity. It's almost not worth acknowledging because who is going to sense the difference between 0.4 THC and 0.6 THC? It's almost no one's, an HPLC is the only thing that's going to detect that, is all I'll say before we jump back to the thing and then maybe go to the conclusions, because I accidentally bumped us down to the, what's it called? Citations. So plant density we already did, ontogeny we're kind of wrapping up, and I think temperature, altitude, CO2 concentration. So there's a lot of stuff around here, kind of lines up with what I've seen in a lot of other research, 25 to 30 Celsius, which is like the high 70s range in Fahrenheit. So, but this was done in northern India. So there are a whole lot of different studies that are evaluating temperature as well as altitude and the impacts that it has on cannabis. So if you are curious, I'm going to just kind of scroll through here. And if anybody on the panel has thoughts or comments they want to stop and check on and see.
Well, let me take a closer look at the first paragraph in this chapter. Okay, yeah, never mind. Curiosity sated.
It's okay. Yeah, it's interesting. I saw that increased density in trichomes and augmented trichomes, which that's an interesting comment there. I'm going to hover over. So if anybody wants to check out the resources there, we've got those. But yeah, this is an interesting one. I know it's different because typically we focus on a single study, and this can kind of be a little bit all over the place. But I think it's just one of our many times that we're touching on scientific research like this and being able to cover so many of the different things, because a lot of this is being studied currently. This one is a 1975 paper, like I was mentioning earlier, Sharma, GK, altitude variation, and leaf. This might have even been the one that was talking about like UV and the different levels at different altitudes. But yeah, very interesting to see them going all the way back.
That third paragraph was pretty interesting too, about the humidity and temperature, how the higher temperature but with lower humidity brought the higher THC percentage. And what's kind of what people are doing now is the higher temperature, higher humidity. So that was kind of contrary to what a lot of people are doing.
It was the old school way of thinking. I think with the HPS, a lot of people really liked to have low RH for one reason or another. I think a lot of people were just trying to avoid powdery mildew and botrytis and different molds. And with limiting air exchange as much as you can to, I think low RH was a safe bet for a lot of people for a long time. And maybe that's the type of stress, just like a drought stress, that maybe the drier RH is in certain points of flower. I don't know if like the entire flower, but I've also seen, I think it can do, cannabis is amazing. Like we covered some of the landrace stuff the other week, and I was showing some of the Afghan stuff that was like in the desert and still doing fine. But then they have stuff in the mountains that's thriving. So it can adapt and do well in tons of different environments. And I think it just produces different phytocannabinoids or different amounts or yields different or looks different. Like you'll see tighter buds or looser buds or, frostier or less frosty, things like that. But ultimately, I think cannabis can kind of survive nearly anywhere, not necessarily everywhere, but if you make the accommodations for it, yeah, it's definitely one of those things that it naturally and with human assistance has gone basically all over the world, which is amazing to see how it's changed and continued to evolve and reproduce. feel like this would have been an interesting one for the American one. He always loves talking about Hawaiian and there's some good tropical climate
Oh yeah.
stuff going on around here. But again, 1975 studies. So they are going back. Chandra et al. This is a more modern one, I think. 2011. So within the last decade almost. This is the one that I often reference, talking about essentially the maximum CO2 levels, where like after you get to a certain point, you're hitting diminished returns. So you're going to be pumping light and CO2 so hard that there's only a certain level that it actually makes sense to do that up to. So trying to see if there's anything else maybe of interesting note before we get into the next section, I guess, is nutritional substrate. We talk about this quite a bit, but it's always good to see the science and maybe touch base there. So I think we'll keep on moving on from the altitude and CO2 stuff, even though we could probably spend more time on there. We're almost an hour in, and Matthew said that he also has a paper that we'll get into maybe in the second hour. So we'll glance through the nutrition and substrate section and see if anybody on the panel has any thoughts or comments on anything that they see. Maybe we could stop and talk, or we can keep on rolling through.
Well, one thing I wanted to bring up here is something we've already talked about before, but I like to stress it. And that's what, 1, 2, 3, fourth paragraph down. The excessive fertilization actually brings THC down in flowers. So everybody that likes to push their plants to the, really high ppms, man, you're bringing your THC down. Maybe that's not a bad thing. Maybe it's not.
Great point.
Oh, it definitely is. I mean, yeah, like
too much love.
It's one of those intuitive traps, right? Like, you mean I can't just supercharge it? No, it's like I mean, it's very simple. It's like unless you're like pumping a lot of iron, maybe you need a little bit more protein, right? Or carbohydrates and things like that potentially. But at the same time, you can't just like eat, 10 steaks every day and expect that like you're gonna turn to Arnold Schwarzenegger.
The hydro store will tell you that, though. They'll tell you, oh man,
course.
pour this
the
bottle
nutrient
on it.
Supplement company will be like, yes, please buy all of our protein creatine.
And just keep on dumping more, do more. The reason it's not yielding well is you don't bottle. You need to buy another bottle to fix the issues from that last bottle. It almost reminds me of another industry that I won't get into, but yeah, it's a shame sometimes. And I understand why a lot of these hydro stores shut down, and poor business practices like that, I think, are one of the main reasons. But yeah,
you can't don't fix your cannabis with peanut butter and trend sandwich. It's just not a good idea, I guess. It's also just waste of resources, right? Which is a critical problem in all agriculture.
Well, and like earlier, we were talking about seeing lots of sources cited. This one has 5. So the others have reported that the increased availability of NPK decreased THC and other phytocannabinoids. 1, 2, 3, 4 studies all found that. 2021, 2021, 2019, and when was this? That can't be right.
2017.
2017. Okay. I was like,
and then
1796, that was the page number.
And then 2 more studies that found the same thing in the leaves.
Yeah. So, I mean, 6 things that show cannabis is essentially, if you give it too much nutrient, you're going to be suffering the consequences of lower phytocannabinoid yield, which is ultimately we're trying to grow phytocannabinoids. That the reason it's called a cannabinoid is because it's from cannabis, and most other plants, for a long time, they thought no other plants were able to produce cannabinoids. So the thing that makes this plant really special. I know everyone loves to talk about the terpenes, and I love terpenes too. I have episode 3 of Greenstock Talks is called Terp Talk or something like that. And I love terpenes as well, but they're found in all plants throughout nature. The thing that makes cannabis different really is the cannabinoids, minor cannabinoids and things like that. But the beautiful ratios that it comes in makes it amazing also. So I'm not a terp hater by any means, but esters, flavonoids, and those things matter too. One of the interesting ones I saw was a study, and it might even be referenced kind of right here, is talking about the different types of nitrogen and how that could increase or decrease phytocannabinoids. So if you're using too much of certain types of nitrogen, I guess maybe Brandon would be an interesting person to talk to about that, because I know you've tried lots of different types of nitrogen and have some thoughts on different nitrogen input sources.
I think I've posted at least one of these papers that are being referenced here, because I think I remember going over this recently myself. Perhaps the DePrado one, because it's a little bit more recent. But was going to say also, oh yeah, and like it's for those who don't already know, I think a lot of people in chat do already know, especially the people who come here every week. I see you, I recognize you, you know, but like mycorrhizae and other microbes won't necessarily, not all of them, but a lot of them that we famously think of, um, those mutualistic relationships, they also have a cost associated with them. And so if the plant's getting all kinds of these nutrients up front, no problem, there's no reason to invest in that relationship necessarily, or at least a reduced reason perhaps. And so in some cases you don't even get that interaction if you're stimulating with a bunch of nutrients in the substrate. So that's a thing to consider as well. I'm not saying that's the reason in these cases, but it could definitely be a contributing factor if you're trying to use these mycorrhizal fungi and other sorts of microbes at the root level.
I wanted to bring up a comment in chat before we got too far away from this actual screen, but the rest of this also states not only the phytocannabinoids, but it also brings down the yield. And WV Grower was asking, is it lower THC because the buds get bigger, so they have less THC by volume? Well, the buds don't get bigger. If you're over fertilizing, you're going to get stress on the plant enough to where it's going to actually decrease yield as well. And it's saying that
Smaller, weaker buds.
Yeah.
It's all bad. Like really, there's no reason to over fertilize your plant. And I know that the Jungle Boys might say, hey, we run 4 EC, but if you don't have the type of cultivation equipment that they have to ensure that things are, and to be honest, I've smoked the Jungle Boys bud, boys. It's not good. And I know a lot of people out there think it's good. Maybe they haven't tried some really good shit yet, but the Jungle Boys product that I've tried, and I've tried a few different ones, it's not even priced at like the good. Maybe they're trying to go for a more value product, and props to them for that. It's definitely not the most expensive product on the market, but these guys that are touting, oh yeah, we run these super high ECs and we use X or Y or Z nutrient at this level. Many of them are sponsored by nutrient companies. So one, they're trying to sell you more nutrients, whether it's, implicit or explicit. That is the case with a lot of them. The reason that they're toting around those things, showing off the Athena or whatever nutrient, it's because they're getting paid for it. And yeah, they're using it, they're growing, it works for them. But so does, Jack's 321 that you can get at the ag store for maybe 10th the cost. Shout out to Brian 420 PM, who has a great breakdown of nutrient cost and ratios on his Instagram. Cool dude, just had a great harvest. Hey, I know he's a long time listener. He might be in the chat right now. So shout out to you, Brian. Cool dude. Maybe someday you'll reconsider coming on the show. I know you talked about maybe wanting to come on and then change your mind, but we'll see in the future. But that being said,
I can comment a little bit on nitrogen and both yield and on cannabinoid production. And so for me, obviously, you guys already know, if people who follow me, amino acids are going to be the number one kind of go-to, and you can get that through a different combination of things, either through the introduction of things like protein hydrolysates, or just from the natural breakdown and decomposition of organic matter, and also through some of the metabolites that are produced by the biology in soil. So those hands down are the best because they have a way of conserving energy. However, this is the real thing with nitrogen, regardless of if it's, amino acids or nitrate or ammonium. You want to be careful about how much nitrogen you start with and how much remains in that system and in the plant itself as that plant progresses through its later stages, because too much nitrogen will both decrease your yield, it will decrease the cannabinoid content, and it'll really, really wreck the flavor. A lot of the harsh flavors that's associated with things that people don't think are rinsed well, it's because there's too much nitrogen in that material still. That's kind of my takeaway with nitrogen. So you want to see nitrogen higher in veg, and you want to see it kind of plateaued downwards. Like it plateaus, it goes up, and then it just slowly decreases throughout the flowering cycle. And you should see natural senescence I know about what 30, 35 to 40 ppm at the end of flowering is okay, but you don't want to be any higher than that.
Yeah, that's like I mean, I don't know, maybe misconception is too harsh of a phrasing, but like I think Dr. Coco has said it too on this show as well. But like, having an extra special, super duper different flowering, nutrient line, like that's sort of not necessary, right? Like, I mean,
Absolutely not necessary.
I've certainly worked with people who have not paid attention to this contrivance and have grown really great stuff. Me, myself included. So, I mean,
you have to keep in mind that plants, there's 3 ways they can uptake nutrients, and then there's only a certain form that they can take them up in. And if we're talking about things like calcium or magnesium, right, it's their divalent form. And then if we're talking about like iron, we're talking about its ferric form. And so when we're talking about these individual molecules, we're talking.
Is there a non-ferric form of iron?
Well, there's oxidized Fe3, like
okay.
If it oxidizes, and that's what happens, it oxidizes in the system. So it needs to be able to stay in that form.
Ferrous form. Yeah. Okay.
Exactly. So in the same thing, when we're talking about phosphate, we're not talking about the amount of phosphorus, because phosphorus isn't available to plant. It's actually the phosphate anion that the plant can take up. And so it's very selective what the plant is actually able to absorb. And so that's one of the things. It doesn't matter. And the plant oftentimes can't differentiate between where that source is coming from.
I see. Yeah. And I mean, you've reminded me that, like, one of the big benefits of a lot of mycorrhizal relationships is this like phosphorus mining that they do. And I want to say it's I want to say it's a phosphorus cation that is often said in literature, but I could be totally off base there. But that could be a difference, right? Because it's going through the fungus first, right? So there's all this transportation cost, right? There's all this energetic metabolism that's being front loaded by the microbiome in that way, which makes it perhaps worth the investment in a lot of cases, right? So that's kind of a great way to sort of put those two things that we're talking about together. I appreciate that point.
There's 2 more sources that are saying excessive fertilization during the growing season can result in decrease in THC concentration and flower yield, number 115 and 140. So that is like up to like 8 now. I think that's a little bit too much fertilizer. Is going to hurt your THC content, which a lot of people are still growing for, as much as people are emphasizing the importance of terpenes and the entourage or other effects in cannabis. THC is definitely one of the prize bonus.
Think about the people who are strictly growing for things like rosin and stuff. These people don't really want biomass. What they're looking for is resin production, cannabinoid production, terpene production, because that's essentially what their end product is, and they're getting rid of the biomass to begin with. So but anyway, hey, I have to it's dinner time here, so I gotta sign off. It was great talking to everybody, and I'll see you guys all next week.
Thanks for joining, Brandon, and have a great week.
Have a good one, Brandon.
I just ordered some of his Amino N Plus at bokashiearthworks.com, and I know he's looking for affiliates. I know Spartan Grown is going to be one, and shout out to Marcus Greenthumb. He's one as well. So if you're looking for, I think, a discount code, you can get hooked up from either of those guys for bokashiearthworks.com. Like I mentioned in past shows, try to pay full price to support my homies' businesses. But if you do want to save that buck, it is the cheap home grow. I don't blame you. Keep that money in your pocket and reinvest it somewhere else. Save that money and check out Spartan Grown or Marcus Greenthumb to get that affiliate link code to maybe save yourself a few bucks on some great products there. And thank you, Brandon, for your time. I know he's gone now, but he's always good to have around when we're talking about some of the science stuff, and appreciate his input.
Yeah, I love that he's got products. So he looks at the science behind it, and then he's got products that's supported by the science. That's what I really like about his page. So when I saw he had an affiliate program, I wanted to jump on board that, because those are products I use in my garden. So yeah, big shout out to Brandon, man. Really appreciate that.
And there's just some more stuff about some of the different types of nitrogen and how it can have different metabolic responses and things like that. But I think we've done a good chunk of talking about the different stations and things like that. There's so much, as you all can see as I'm scrolling, there are literally hundreds of studies. So we could talk about this for hours and hours and hours. So in the sake of brevity, we're going to talk a little bit about water availability before we move on to the final thoughts and conclusions, or just mention that this is pretty much exclusively talking about messing around with drought and examining different turgor pressure and things within the plant. So a lot of stuff that we've actually talked about in the past. So I don't want to spend too much time on it, but the final part before we pass it over to Matthew's paper is the conclusions and future directions, which I think is worth potentially reading over. There's just 3 paragraphs, and then we could give our own input on some of the stuff that we read and thought about, and then move on from there. But it says international narcotic conventions and associations legalization have constrained the establishment, characterization, and use of cannabis genetic resource collections, which is very true. This has resulted in the underutilization of the gene pool, variability, and cultivar development. Moreover, a limited amount of research on the influence of environmental conditions on phytocannabinoid yield is evident. Prohibitionist legalization has discouraged scientists from studying cannabis production, the main factor responsible for the current lack of research in several countries. Legalization is changing due to recognition of medicinal and agricultural value of cannabis plants. I've cotton off, but we have 2 more paragraphs. If maybe Spartan, you want to take the next one, and then I'll tap in for the final.
In recent years, the number of works on the topics covered in the review has increased significantly. However, they are not enough to determine the influence of management techniques in abiotic factors or phytocannabinoid production. Thus, it is necessary to develop basic research to evaluate how nutritional availability, water availability, harvest time, CO2 concentration, light intensity and quality, different types of stress, pruning, and other management methodologies could ultimately influence the phytocannabinoid yields for different genotypes. Epigenetic and biotechnology tools, such as omics-based methods, can explain how these compounds are synthesized according to the physiological responses under crop management techniques and different environmental conditions.
And I'll actually pass it, Matthew. Would you be okay with reading the last one, or should I take it?
I can take it. Additionally, the fibers produced by plants grown for the production of phytocannabinoids can be used as raw material for several industries. However, there is little research on the production of fibers and phytocannabinoids. There is a need to explore the concomitant productive potential of these raw materials to ease circular bioeconomy practices. Detailed knowledge of the biological and physiological processes of cannabis sativa plants is important in order to support the development of efficient cultivation methods, aiming to provide higher yields and consistent quality.
Thank you guys for reading that. As we do have a few blind growers out there who are listeners of the show and people that listen to the podcast, it's nice for us to at least be able to read off those last few paragraphs to kind of give a summary of how they feel about what they've gone over in this paper, which is literally hundreds of different papers that are all, maybe not hundreds, let's see. Yeah, it's hundreds, 157 different.
157.
That's a lot of
Damn.
research that they went through, a lot of time that they put into this paper for us to be able to, in an hour and 15 minutes, quickly glance through it. Obviously, we're not going to be able to touch on all of the detail of all this, but I will be linking this in the show notes, both on the podcast and on the YouTube. So if you want to go through and look at it in more detail, you're more than welcome to. And I just want to say thank you to everybody for hanging in there with us. We got 104 live with us in the chat. These science talks can be a little bit dry at times because we're looking at a share screen with lots of text, but I like to try and bounce it off the group and talk a little bit with the chat overall. And I tend to have good times on these shows and we get a lot of positive feedback. When I did a poll on the Spotify, only 10 people answered, but 4 of the 10 said that they wanted more science research article paper reviews. Then I did like an A, B, C, and D. It was like more science was A. B was like chat Q&A and chat guests. C, I think, was like general grow topics. And then D was a mix of all 3. And it was like 40%, 20%, 20%, 20%. So I think everybody wants a little bit of everything because those are like the 3 main types of shows that we do. And I like doing the mix of the 3. So it's fun to kind of bounce back and forth and not have to do one type every single time and be fixed into one thing. But yeah, does anybody have any general thoughts or conclusions? As we read the scientists kind of really wordy conclusions on what we went over. But I'm curious if you guys have any thoughts on what I'd consider at least one of the meta-analysis that did a pretty decent job summing up the general state of affairs in science around cannabis that we know today, in regards to abiotic factors, that is.
Well, I would say, I mean, I'm glad that I'm very glad to see the interest in that. Of course, like kind of obvious, a lot of crops have to deal with these factors. It's very important to do that kind of research. And also, we're in an interesting time. I guess we're in an interesting time in plant and sort of agricultural research in general, because we have the ability to sort of synthesize so many different kinds of factors together at the same time, that we are kind of moving into that it's an interesting sort of criticism that I sometimes hear about research, which is, I don't think, very accurate a lot of the time. Sometimes it is true, but not all the time, which is that, like, oh, scientists, they're always trying to like control for variables and only look at one thing when the world is so diverse. So the research is totally invalid. And it's not true because a lot of times, yeah, sure, some research is just looking at one particular variable, but like you said, Jack, a lot of this research is then replicated, or they add other variables. And but you kind of have to know a little bit about what you're talking about first and kind of isolate in that way, and then you kind of bring it together. But the idea that like researchers just don't have like a holistic perspective is really uninformed. And as you can see, we're able to take a look at a lot of those factors, even with something like cannabis. So I'm excited to see where that goes.
You and me both. I really hope to see more stuff get studied. But Spartan, did you have any thoughts on that?
Yeah, I'm just kind of the same mind of Matthew. I'm just encouraged, honestly, to see some stuff like this. I'm always, honestly, every time I see meta-analysis, I'm already half disinterested, because they're not really, I mean, they're looking at other people's work that who knows, they don't have, I don't feel they have the time to really dig into each study and see how the methods were and to see how valid they were and at the time and all that stuff. So a meta-analysis, I take it as a kind of like an average, an average kind of thing. And they are cool to dig into. And this particular one was nice because of how wide scope it was. And it's just a drop in the bucket of how many, it illustrates, like it's a drop in the bucket how many different factors can affect the cannabis plant, and why the most common answer you're going to get when asked a question about growing cannabis is going to be, it depends, because there's so many factors. So I love that the science is being done. Is, I look at something like that and I feel almost like throwing my hands up and like, what's the point, because there's so many factors. But at the same time, science is what makes science so great, is that it tackles things like that to the minutia. So one group might just take one cannabinoid and research that to the nth, you know what I mean? And that's what makes the humans so great, is we can communicate that with each other, and we can divide up our time and figure this shit out, even though it seems like it's impossible. So we just have to give the guys time. And I think the conclusions brought up a really great point about regulations. The regulations have been standing in the way for so long of the science of it. And as these regulations fall, which they have been, it's an exciting time to see the science and to really see where this thing goes, because wow, look how far we've come without the science. I'm excited to see where we go with the science.
And so often our criticisms of the science are like, oh, maybe they didn't do enough plants, or maybe the plants weren't looking good enough, and maybe it's because they haven't been cultivating for long enough, because it hasn't been legal for long enough. So you're dealing with a lot of brand new growers that are also scientists. They're professional scientists. They're very, very good at the science, but the cultivation, as we talked about in the practice of growing episode, is different than knowing what to do. Actually getting it done is challenging. It's a different set of skills. It's more of a farm laborer type work than a scientifically minded type of work. It can be. You can set up automatic watering like we saw with stairs at plants, and you can do things like that. But at the larger agronomic scale, it becomes like most other agricultural products. I mean, you can grow it in fields, but you can also grow it in small greenhouses or in a tiny closet. So it's got tons of variability, and it's exciting to me as well. And the thing that I liked about this meta-analysis was, unlike some that will be like, ivermectin doesn't work because these 100 studies said this about that or whatever. This one was like, look at all these abiotic factors that exist. Here are all of the studies about them. So it wasn't trying to say like, this is the claim that we're making, or like, this is the extrapolation we're getting. It's like, here is the pool of research that falls under this categorization. And if you want to go dig deeper into it and find out, like you're saying, Spartan, spend the time yourself and dig into, is this, because like some of those things we looked at, I'm like, this sounds a little fishy. Like I would like to look into that more. And I probably will after this. I'll go back and listen to them and be like, man, I was listening to that. That didn't sound right. And I'll look into the study and be like, something was fishy with that study. And maybe it didn't replicate, or maybe there was some weird genetic that they used that maybe caused some outlier that we would never come across because none of us are running with that genetic or whatever it is. So there's a lot of factors, like you said, but I like that once people start to, like you said, become the specialists and they narrow it down and they know best temperatures, these are the best humidities, these are the best elevations, CO2s, light types, and genetics, all that different stuff, then we can kind of dial in for what you want, because there's not like a best, right? It's a best for what? The best thing to produce THC is not the same thing as the best thing to produce CBD. So those are two big goals with cannabis, right? So if I was trying to produce the most THC and you're trying to produce the most CBD, we're definitely going to be taking different genetic paths, even if our cultivation is identical. So knowing what you're going to do going in is a major factor, and then what you're trying to get out of it. And even the scientists are kind of at times not struggling with that, but having to address that, like, who is the target audience? Like, I think they're doing a lot of this research to work with industrial cultivators because they're the ones who are going to be, paying and benefiting from this types of research. So
I think it's going to be a consolidation in the market. Once we get a mature market, there's probably going to be 5-10 popular cultivars that are grown massively, all because they perform well or they're designed to perform well. But oh man, we got these chat pots. They're so annoying. I hope I hit the right one. I did.
It's a sign that we're doing well. We're popular enough that some algorithm wants to send them over to us, but I appreciate that you and the other moderators are good at keeping their eye out to knock those people out of the chat, because no one really wants to see them or accidentally click on any of their links. You were saying something, Spartan. I don't know if you still have your thought. I kind of lost it.
I lost my thought. I don't remember what I was talking about now. Sorry, I was cleaning up the chat.
It's okay. Noah the Grower, do you have any thoughts before maybe Matthew? I think you said you had another paper that we wanted to pull up.
Yeah, I do. I'm ready on standby.
No, I've been just listening. So a lot of this stuff is kind of new information to me. It's earlier that conversation about plant count and bigger plants versus smaller plants. That's always kind of been like a pet peeve of mine. Just like back in the day, it's a little different now, but back in the day, it was like guys had 1,000-watt hoods, HDI light, high density lights, and they would always try and get like a pound of light, and they'd always go like a 20-gallon pot or whatever, and they would get. But I would always try and do like 4 or 5-gallon pots, and I would always hit like 23 ounces. And they were like, "How do you do it?" I'm like, "I just like the different, different sizes," and it was always just something I did back in the day,
So like more plants per hood than doing like one massive plant. So you're splitting up the amount of yield on each plant is kind of what you're saying?
Yeah. Well, just, what I mean? Like, and that's what I would always say, we'll get more grams per watt. That was kind of what it was, with the, 1,000-watt lights and stuff. So that was always just kind of trying to get as many grams per watt as you could. And, also just at the same time trying to have quality, too. So.
Yeah, definitely. Quality is, at the end of the day, if nobody wants to smoke it, then why grow it? But I think, Matthew, if you're ready to go ahead and bring up that article, we could jump into that one next, because we got about 20 minutes left before Spartan Grown heads on over to the Michigan Bros Grow Show.
Alrighty. Do I have access and everything?
Do I have permission?
I'm pretty good. I've been setting it at the beginning of each show, and I just double checked, and it says multiple participants can share simultaneously.
Oh, very nice. Very nice. Now I'll just, well, that's weird. All
Can
right.
you preface the article maybe and give us
Yeah. So basically, yeah, I have it right here. So you all can see it right now.
Bio 1 complete.
Yep.
Yeah. So I've been working on a in-depth presentation for powdery mildew. So I thought I would just share a really cool paper about it that I've actually posted several times or referenced it several times in my social media because I like it so dang much. Because it has really great diagrams. So it makes things that are kind of complicated, like you said, Jack, very dry. Even for somebody like me who likes to read about this kind of stuff, it can be a little bit, of a siege. So the diagrams are kind of nice. So this paper is called Biotrophy at its Best, Novel Findings and Unsolved Mysteries of the Arabidopsis powdery mildew pathosystem. So it's not cannabis, but Arabidopsis or Thale cress is a model organism that we use in research for figuring things out about other plants, because a lot of things that are true for it are also true for, again, many other plants. Not everything, and there are going to be unique changes, of course. That's why you do specific research, obviously, but you might preliminarily look at things this way. So again, like I said, my favorite thing about this research is that it gives you a great overview of powdery mildew, but pressed for time, we're not going to get into a lot of those details. I do want to focus just on the pretty pictures. So it goes over how powdery mildew is classified a little bit, also a little bit about its biology, and then kind of what it does. And so in the smallest way, or the most simplest way I could say, is that powdery mildew is like, it's a fungus that it's most of its entirety, like 6 out of 10 genes in its genome are devoted to basically taking over a plant cell, hopefully multiple times, ideally. But that little sporling, that little sprouted spore that gets on your plant, it doesn't have a lot of options. It has to basically try against a cell once, maybe twice, maybe. And if it doesn't succeed, it's dead, it's toast. And so all of the spores that are around right now are from an unbroken line from 100 million years ago or so, of powdery mildew that we're able to get by and get going. And they even predate, of course, at 100 million years, they predate the asteroid that wiped out the dinosaurs. So they're doing something right. So here's this diagram. I really like this because it kind of shows what's going on here in a powdery mildew infection. So basically, powdery mildew spore, which I'll represent here, I think, it makes this little hypha, this little structure, which we call an aprosorium, specifically it becomes the aprosorium. And then as it penetrates through the plant, it makes this big bulb here called hostorium. And the thing about it is that you gotta know this too about powdery mildew, which I'll go over in presentation. But it actually has been found recently that it evolved from, or belongs in a group of fungi that contains mycorrhizae, that contains other beneficial, quote unquote, or sort of like mutualistic fungi for plants. And also a lot of saprophytes, a lot of fungi that break down dead plant matter. And it's thought that powdery mildew evolved from a line of fungi that broke plants down and then acquired traits over time that allowed it to actually go on to colonize living plants also. So it seems to use a lot of the traits that like good guys like mycorrhizae use to colonize the plant. So powdery mildew is like the nega mycorrhiza. It's the anti mycorrhiza. It takes nutrients from the plant instead of giving it to the plant. So this bulb here, it fuses with the membrane of the cell. So you got the cell wall it bores through, but then there's this other thin membrane and it fuses to it. And when it does that, you see when it's penetrating through the cell, all of the various organelles and little cell structures inside the cell, they actually orient to the place where the powdery mildew's tendril, its penetration peg, is going through. And they're there because they have genes that alert it when there's like this sort of physical pressure. Because when a mycorrhiza goes through into it, you know, it wants to be there to be able to facilitate that process. Unfortunately, that's not what happens. So what this hostorium is it's where all the nutrients are going to go, but it also dumps all of these things called effectors, which are like proteins, and you might even classify some things like toxins in this light, but really usually there's proteins that are going to like basically blind and stupefy the plant's ability to suppress it. It's going to do the suppressing before the plant can suppress it, essentially, and it will reprogram how the plant cell expresses its genes, which is, you know, pretty significant. Now, if the plant is able to recognize this for what it is in the very beginning of the infection, which like a lot of resistant plants do, then that thing never gets to that point, it dies. That's really good resistance. But sometimes what happens is it produces these things called, and I'll finally go to another diagram here. Yeah, this shows kind of how it's penetrating here. We got these cool little I think it's a scanning electron microscope, I want to say, or fluorescence or something at least. But yeah, this is oppressorium. There's the little hostorium. This is 6 hours past infection, 12 to 14 hours past infection, 24 to 48 hours. You got this one infection, and they're going on to another cell. 3 to 7 days. So you usually get only a few cells after a few days. So the very beginning parts of powdery mildew infection, are kind of small and invisible. When you get into this this plant cell, they're producing all of these little things called multicellular vesicular bodies, and they're just loaded. They're just packed with all these proteins that are just going to be shipped off and suppress the immune system of the cell. They bind with things that are supposed to be toxic, break them apart. They basically tell the plant to stop producing some of these defenses altogether. They might even tell the cell to produce signals that will interfere with the signaling that it would use to tell other cells, hey, hello, we have a powdery mildew problem. You should prime yourself, for resistance.
Is the resistance to the plant act like it's almost like with a virus where it tries to oxidize it, or how does it try?
Yes.
Okay.
Yeah. So that's called hypersensitive response, and there's actually a few different ways that it happens. And some
you bring it right now. It says oxidative burst right there. I didn't see it.
Yeah, you're right there. Yeah, you're there. You're correct. This is admittedly, this is how a lot of cells deal with their problems. Is by taking it with them. But that being said, some biologists, some people who are way more learned than I am, they call it certain ways that this is triggered, sort of a different term. But for our audience, I'll just say that it's all hypersensitive response. But basically, yeah, what it will do, even with plants that can't totally resist the fungus, like in the other example, they tend to have some generalized responses to things that get in that shouldn't be there. And one of them is kind of like what white blood cells do. You might know from your biology classes that white blood cells come in and they envelop like a bacteria or some other thing that shouldn't be there, and they eat it to death. They phagocytose it and kill it. And so something very similar happens here, where not white blood cells, but other proteins, which we can see here on the green section. This is called resistance to powdery mildew 8 and 8.2, I think, are these particular vesicles. But yeah, that's literally the name of the gene that's promoting this effect, is resistance to powdery mildew 8. so presumably there's 1 through 7, right? But it detaches, it reattaches onto this bulb, the hostorium, and over time, hopefully, in a particularly robust resistant case, you do 2 things when you do this. One is you shut off the powdery mildew's ability to dump those proteins, and the second thing you do is you physically interfere with its ability to absorb water and nutrients from the plant cell. So it's really kind of an all-in-one package. And so hopefully you're able to encase it totally, shut it off from the nutrients, and then you produce massive amounts of these reactive oxygen species, which are often used as signals also, but it will also just basically destroy everything in the cell and deny it to the enemy. And then if it's the first time a spore is germinating, then this is basically curtains for the spore. If it's the case where the spore's already got a few hostoria, then maybe this is just going to make it a very uphill battle for the powdery mildew, which could result in its death a number of ways. Maybe other cells get primed and then those new ones get destroyed and it just lived for a small period of time, but not very long. Or it might be able to live a longer period of time, but other factors, like we said with the environment, maybe it's very hot, maybe it's very intense UV radiation. The fungus doesn't like that either. So if it's having a really devil of a time, even if it was in better conditions, maybe it could still contend with the plant, right? But the high resistance plus, UV radiation plus other things going on, just might be too much for it, and it dies that way. So it's very important when we talk about resistance to both acknowledge that there are different kinds of resistances that do different things, even to the same target, but also, there's all kinds of things happening at once in real life. So if you're trying to judge something for its resistance, you kind of have to control for those variables, like we were saying earlier. And this B section here, this is showing, so red, the propidium iodide, that's the colorant, is showing plant and fungal structure. Did I miss something? Micrograph of the GFP. No, that's also a colorant.
Are we doing inoculation trials anywhere yet, Matthew? Is it the case where they're taking like a cultivar and saying, all right, we inoculated this with powdery mildew, we know it has it, and then we can see either type within that cultivar has resistance, or if they try 10 cultivars and one of them doesn't have it and the other 9 do, you could imply that, well, it was inoculated with it, so if it doesn't have it, then it's got resistance.
Yeah, there are groups that are doing it. I feel like a lot of people who breed, I've had a conversation recently with somebody who took great umbrage with the suggestion that, they should be controlling for environmental circumstances with their resistance trials, or claiming that their plant is resistant. But like Cornell, I saw a paper there where they were doing inoculation tests for hop powdery mildew, for example, and other powdery mildews as well. I'm pretty sure that the University of Guelph did a study regarding that. I know that there are also private researchers who are probably doing this as well. And in other agricultural spheres, this is par for the course. You would absolutely be doing inoculation trials to at least tell, like, you can't just say like, hey, these plants over here had powdery mildew and these plants over here didn't get it, and they're pretty close together. So if this one didn't get it, then it must be resistant. Like, that could be true, but you don't know for sure. And a really easy way to do it that doesn't cost a lot of money is to actually try to get them infected.
Rub the leaf on the leaf.
Rub the leaf on the leaf, yeah, and document that. And if it doesn't colonize, then that's pretty impressive, because there should be thousands of spores on like a really patchy cannabis leaf of sufficient size. So I mean,
this picture right now, that's giving me skin crawl. That is a lot of powdery mildew.
Yeah, exactly. Like if your leaf looks like this, and you put it on the, yeah, you're in trouble. But if you take the Col 0 over here on the left, and if you were to take one of those leaves and put it on this MLO mutant, which I assume doesn't have the MLO genes, because they're talking about, we can get into that though, but basically if you do it and it still remains like this, that's what we might call in the business robust resistance. So, but pathogens can mutate and plants can mutate too. And the other side of the coin is that the powdery mildew gets a vote. It's constantly mutating. All these different spores that it makes are going to have small little mutations that may or may not even defeat robust resistances. On the same token, a plant that you might breed with, as you're breeding it, if you're not checking to make sure it retained the resistance, right? You can't just assume, because obviously the plants that we had millennia ago might have been more resistant, which many times is the case. And what we're finding when we go and introgress landrace, right, and other feral populations and things like that. Well, so obviously when we went and we bred for other traits, we lost those resistance traits, so they don't stay either necessarily.
We didn't know or even think to pick for it in many cases because people were trying to cultivate in environments that were suboptimal for the PM to be there. So the cultivars in a perfect environment, quote unquote, aren't going to have to worry about being resistant. So many people never selected for it. So that's just the sad reality of the weaker genetics that might be prettier or more potent or taste better or get you more high, but they might not have the same resistance to powdery mildew.
Yeah, exactly. And in the research I've done for this presentation, I learned that hot powdery mildew was a massive problem in the Pacific Northwest, and basically caused, just like it did in New York in around 1997, 8 square kilometers of hop production just basically was gone, destroyed, in a place where 95% plus of hop production happens in the US, right?
That's scary.
So that was, yeah, exactly. So we should really consider that this can be a massive problem for cannabis as well. And the MLO genes, for people who were curious, that stands for mildew locus O. I talk about it a lot, but I'm also asked about it a lot from newer people, and I don't know who's in the audience. So I want to say that basically, like I said before earlier, powdery mildew learned a lot of its tricks from its brethren, its mycorrhizae brethren, and other things like that. So it exploits the same responses the plant has to allow the good guys in. It pretends to be them, and then when it gets its foot in the door, then it pocket sands the plant cell responders so that they don't know what's going on. And then it takes all of those nutrients and grows stronger. And so when you get rid of some of those genes that allow for the plant to facilitate that interaction that it would have with mycorrhizae, the powdery mildew basically can't even interact with the plant, essentially, in the way that that's supposed to work. And so that also creates resistance by a gene not being there, or a group of genes not being there, which is kind of interesting to consider too. Those are called susceptibility genes, but it's kind of a misnomer because it makes them susceptible to mycorrhizae, which you want as well. So yeah, it's all complicated.
Double-edged sword, right? So to speak.
Yeah, exactly. Right.
Some clones I heard reported by a guy who works with soil sampling. He saw the same clone grown in a greenhouse where one was covered in powdery mildew and the one right next to it that the powdery mildew plant died and collapsed onto, even though it was the same exact genetic, the other one didn't get PM. And when he sampled the soil, there was actinomycetes or something, different levels of that. And the phylosphere generally was much healthier in the plant that didn't have PM versus the one that did have it. So even on the same genetic, if the phylosphere and soil conditions aren't proper, it can be more susceptible to PM because cultivation errors and things like that. But before I pass it back to you, Matthew, I want to give Spartan Grown a chance to give his final thoughts and shout out before he goes to the Michigan Bros Grow Show and we wrap up this paper.
Yeah. My only thoughts that I had to add was I was going to ask Matthew a question about if he'd seen anything about the nutritional aspects of if the plants had more silica available. I've heard through Harley Smith, I think his last name is, from NPK Nutrients or Industries, NPK Industries, that if the plant has sufficient silica, that it'll build more of almost like a pectin-like substance instead of water substance between cell walls, and that it really makes it hard for powdery mildew hyphae to penetrate to even pull up any water. but he didn't cite a paper or anything. It was just his own research. So I was just curious if you'd seen anything to substantiate any of that. But other than that, I want to shout out the chat and everybody in chat that comes to show up every week. I mean, it's almost like a family. I'm seeing the same names again and again, and I feel everybody on the panel is family to me. So it's always great to come on and catch up with everybody. And I really like this episode, and I really do like digging through these articles. I just wish that we had a little bit more time to go through before the show, but I got to a good. I did some speed reading and picked out a few things that really stood out to me. But it was cool. I like learning, and I definitely learned a lot from just that one research paper there, which kind of goes over a bunch of research papers. Anyways, thanks everybody, and I just think when all said and done, I think the best thing we can do is grow our own cannabis at home. And that's if you're doing that, I think you're going a long ways to doing the right thing when it comes to cannabis. So keep growing, everybody, and I'll see you guys in a little bit here over at the Michigan Grows Grow Show. Love y'all. I'm out of here.
Later, man.
Spartan. Thank you so much for coming. Always great to have you.
Yeah, I do have an answer to that question. Basically, silicon and some compounds with silicon in it. There's that robustness of the barrier that I've read about, but also I guess it can be a pretty potent has signaling capabilities, at least in certain forms. Apparently that was actually pretty interesting to read about. I am going to be putting that in a presentation too, but I still need to look into that particular aspect a little bit more. But I'm very excited to see these kinds of I don't know, like these sort of newer or perhaps like less intuitive ways, because the physicality makes sense because like we think like sand, right? But like but having them being used in that way is kind of interesting. Here, I actually accidentally left the screen. So here, I just have one small section left, basically. So this figure 8 here is kind of interesting. Basically, with it, I want to explain that there's like, because we mentioned these signaling profiles, right? So like, what are the ways that the plant can prime itself? Well, plants have finite resources and they can't like move away from their threats. So the way that they sort of outdo their competition is they either outgrow it, or they have something toxic about them, or they have something like with their signals that they have to actually use to figure out how much of a resource they're going to use, how intense a particular stressor is that they have to use to mitigate it. Because if they put all of their energy into one stressor and another stressor comes around, that's not going to be very stable. So plant researchers have found that plants have a very sophisticated way of doing this. It's also similar in our own bodies. We have these immune signal response agents. And so if it's not stimulated, then it's not primed, right? But then certain things happen, like, the powdery mildew here, it penetrates into the plant, and perhaps something in the cell recognizes the chitin, right, in the cell structure of the fungus, right? So fungal cells have chitin, and so that will trigger a response. So it will for like insects, for example. They put their mouth part into the tissue, and they might have saliva that affects this, right? That keeps the plant from recognizing it or recognizing it substantially. But if they do happen to come across it, then there will be this massive signaling. So there are aspects of the physical structure of the powdery mildew or the chemicals that they produce that are recognized, and then that stimulates a response. That's one way. Another way is from, as I see in the diagram, we have a mycorrhizal fungus, is Piriforma spora indica. So it being associated with the root microbiome will prime the production of certain compounds. And you might think to yourself that that's because, this fungus is like letting them know, or like, massaging the plant's muscles, so to speak, metaphorically. But I'm not altogether sure that's what's happening. I think that you could interpret it that way, but it could very well be that, because the immune system regulates the good guys too, so they don't become bad guys, or they don't end up going somewhere they shouldn't. There's various microbes in your own body that are great where they are, but if they got into your blood, it would be a bad day, perhaps perhaps a bad life. So the same thing happens in plants too. And I wonder if there might be some of that. The presence is merely causing this uptick in immune response to be induced, essentially. And then the other way you would have this happen, so there's a genetic factor or a microbe that has genes that does this, but then you also have yourself as a cultivator. You might apply like jasmonic acid, which is what the JA stands for. The JA is jasmonic acid, the ET is ethylene, and the SA is salicylic acid. And so some signals are more associated with certain stressors or certain targets, certain organisms, and others with others. If you pump the wrong signal for the wrong situation, that can be incredibly bad for the plant. So that's why, again, you can't just say that something's resistant, or, oh, we'll just upregulate this immune response compound. Everything will be fine. It'll prime the immune system. But if you prime it the wrong way, that's a bad day. And so, again, I just want to really emphasize that is not necessarily going to be helpful for you. It can be very hurtful.
Did you find? Sorry to cut you
yeah.
off, but Smart mentioned Harley, I think from NPK, and I think that he also mentioned something about like using biostimulant effects of something like humic or fulvic acid to help potentially mitigate or prevent powdery mildew. And in your research, have you come across any research substantiate that maybe humic or fulvics have some way of potentially mitigating or preventing powdery mildew?
Yes, I definitely have, but on the top of my head, I can't remember a lot of details. And to sort of, that's a very good point to make right here while I was ranting about, immune system pathways and priming, because, I've just said in one breath that you have to be very careful, but on the other side, there are different compounds that do different things and sometimes multiple things. And sometimes even botanical research doesn't necessarily know all of the details. So like, I do want to give that caveat, but that some compounds, you might be able to get away with a more generalized response, and it might be a safer option. Whereas if you like really uptick one particular compound, then it can be a lot more of this sort of like detrimental effect, because it could even cause like something really hasty, like that causes like the plant's cells to lyse themselves or something like that. So
The dose can be the poison sometimes.
exactly the dose is the poison.
You're right, yeah, and then in some cases maybe it could accelerate versus prevent infection, depending on what stage maybe the infection is in and things like that.
Yeah, like I had a conversation over social media recently where I was trying to explain that, as a biotroph, as a powdery mildew it can only interact with living plant material. It can't grow on dead plant material. It has to be growing on a plant that has its living cells that are getting sugars that it feeds on, and, other nutrients that it needs.
Which is bizarre, because
survive.
didn't you say it evolved from only
Yeah.
being on dead material?
Yeah, that's the estimation anyways. That's the idea. And it's a good question, because the real, like, one of the traits that it has that lends credence to this understanding is that particularly it has pectinases, or cellulases that it shares with other, like, a very similar phylogenetically related organisms. And so the chitinase would probably like the first helpful thing, which is to just bore through the plant cells. And it's possible that powdery mildew in its earliest days, or the ancestral powdery mildew, maybe it was a lot more destructive than it is now. Like botrytis, for example, is it's a hemibiotrope. It can go biotrophy or necrotrophy. And so what it does is it will manipulate the plant. See, plants can't win. This sophisticated, hypersensitive response that kills the powdery mildew and also viruses, it's not very good for botrytis, because botrytis literally triggers it on purpose, makes it think that, or in other ways, induces it inappropriately. So the plant will have its cell destroy itself, and then the botrytis is like, thanks for all of this nutrient matter that I'm now going to feed on.
So damned if you do, damned if you don't.
Exactly.
in some sense. One other question or comment I've heard this reference a few times by Breeder Steve. He says 35 degrees Celsius or 95 degrees Fahrenheit. And I believe he does 10 minutes per day for like several days in the first week. And then after that, he can lighten it up to maybe like 10 minutes or like 30 minutes a day for 1 day a week. But he's saying because it doesn't survive over that temperature, if he gets his greenhouses up to like, let's say 35 to 40 Celsius or like 95 to 100 Fahrenheit, that it will literally kill off the powdery mildew. So I'm curious if you have any thoughts on that as far as a cultivation practice, and is it something that would be feasible and implementable, and is that even actually true that it would potentially work?
I haven't experienced that, and I haven't read any research that supports that. I do know that one of the things about powdery mildew that's sort of unique is the spores have a large proportion of them containing water. And I guess because a lot of spores, require moisture to germinate, like just in the group fungi. So, it does really good or better in like arid conditions to germinate and have greater success that way is the implication. As far as humidity is concerned, I mean, temperature, I only can think of in solarization, the UV radiation having a negative impact on the spores. Also, the powdery mildew doesn't do very well in like full moisture, like 100% moisture, like wet leaves.
Sprayed it, yeah.
It will drown the spores. The spores will drown.
Which is like counterintuitive as all get out.
Yeah.
You always think moisture is going to cause powdery mildew, but then actually it will kill it if you completely submerge it for a certain period of time. So very interesting.
Yeah.
Do you have any final thoughts to wrap this paper up, and then we'll close it down?
Just a penny for your thoughts. One other thing that powdery mildew does that most people have never heard about is that will cause cells to replicate their genome and become polyploid. And this is thought to basically increase all of the good genes that will give the powdery mildew the nutrients it wants, essentially. And so that's kind of cool that people have never heard of before, usually when I talk about this.
Didn't even know that was possible.
Yeah.
At all.
So there you go. That's another cool and
tip. Terrifying.
Terrifying too.
Terrifying facts about powdery mildew.
Exactly. That's right.
All right. Well, Matthew, where can the people find you?
So you can find the presentation that I'm going to be doing on the Future Cannabis Project's 02 channel, FCPO2. That will hopefully be on the 25th. And you can find my work, which is often this kind of entertaining stuff, on my YouTube channel, Zenthanol. You can professionally inquire about my work if you need help, consulting help, on zenthanol.com. And you can find me on social media websites, Twitter and Instagram at SyncAngel.
Thank you so much. And next up, we've got Noah the Grower.
Yeah, it was an interesting conversation today. I'm always learning new stuff. I'm Noah the Grower with 2 E's on Instagram. You can find me there, and see everybody next week.
Looking forward to seeing you and happy to have you back. Hopefully everything's going well. And it is kind of interesting that this is like pretty educational content, I would say, at least. They're as educational as we can make it, but it's also I guess something that people are entertained by. So that's a good double whammy there, that we could keep people hanging in here for 2 hours. We've had roughly about 100 people the whole night, and it's great to see all of you. Thank you so much for coming. If you want to find me, I'm at Jack Greenstock on Cannabis as well as Instagram. Jack_Greenstock is my backup account there, and my name on Twitter. If you want to email me, jackgreenstock47@gmail.com. And if you want a copy of the book, 50 Strains of Green, you could go to 50strains.com. I'm going to do the pre-order for 50 Strains of Purple once I've got like dummy copy myself from the printer. I want to make sure it's all done, finished, and everything before I start taking pre-orders. and then I'll get the prints going and sign and ship them. But with that being said, thank you all so much for coming. And for Dr. MJ, grower love, everyone. And we will see you all next week. Peace and love, y'all.
Keep growing.